activity of tropical rainforest trees. And I think I've spoken to most of the speakers here, but I'm going to give you a 5-minute warning when you're 8 minutes through your talk, and then a 1 minute when you're 12 minutes through your 30-minute talk. Great, thanks very much, Sarah. Now I'm going to welcome Ellen Fry to speak to us about plant functional traits inform ecosystem functioning and resistance to drought on a calcareous grassland. Thanks, Ellen.
Hello. I've come to talk to you today about my work on the Wessex BEST Consortium project. My remit into how plant functional traits... What's happened? Okay, we're back. To look into how plant functional traits can be used in calcareous grassland restoration and how drought resilience and resistance can be affected by trait choice and species choice. This experiment's gone on for 4 years now, but I'm only really going to talk to you about 2015, which is when we imposed the drought for the first time. So the work took place on the Wessex landscape, which is Salisbury Plain in the southwest of England. You can see in the little map in this, in the top left corner. The Salisbury Plain is the biggest area of sort of unbroken chalk grassland in Europe, I'm fairly sure, I think. And it's pretty unique for a number of reasons. Chalk is very different to other types of grassland. The soil is incredibly shallow and rocky and horrible. There's hardly any soil organic matter. The nutrient load is very, very low. And it's subject to an enormous number of pressures. And you can see here Stonehenge in the top right corner. You've got the Ministry of Defence. And so much tourism, it's unbelievable. But if you leave these grasslands alone, and the species-rich incarnation, if you look on the bottom left corner, it's some of our most diverse grassland in Europe, and you can have up to 40 or 50 species per square metre of grassland plant, which is incredible, really. So it needs to be protected. A lot of it's very highly degraded. You know, if you get nitrogen deposition and so on from airports and whatever, then it's going to degrade these grasslands very, very quickly. So we were interested in restoring an area of chalk grassland that sort of started off in looking like this bottom right corner, and it had been arable farmed for about 20 years, and we were wondering if we could use species choice based on plant functional traits to increase soil organic matter and create a stable grassland sward that's not losing loads of carbon and loads of nitrogen to mineralization, so everything's being retained in the system. And we were wondering what the best combination of plants and traits were to do this. So I'm interested in roots primarily. They're very underrepresented in the literature. They're incredibly hard to measure in situ, obviously, they're underground. And if you're doing a 4-year study like this, you can't turn up and keep digging it up every time. So what I wanted to do was to find some root traits that were something that you could kind of understand based on what you can see above ground. So things that aren't going to change, like specific root length and so on, which is very subjective. And I wanted it to be accessible to farmers and practitioners and so on that, you know, don't have this kind of level of, you know, maybe functional trait knowledge that, you know, we do as academics. So I wanted it to be sort of applicable across a number of different scenarios and different sort of people doing this work. So I had a bit of a think, and basically what I wanted to do was to find in the trait databases, so there's freely available trait databases all over the place, Ecoflora, Leda, not so much Tri, but, you know, Grimes' book has all sorts of traits as well. And I thought maybe I can do something with categorical traits. So I came up with some simple hypotheses based on very simple root architectural classes. And this is a really simplistic version of what I came up with. But you know, on a sort of basic level, you've got tap roots, which are very common in chalk grassland species. And they've got a low surface area to volume ratio, which might mean that there's less opportunity for microbes to take hold, to create an environment, because there's less sphere of influence around taprooted, potentially, than these plants on the right, which are the fibrous rooted. So, and this could mean lower processing rates, lower decomposition, lower mineralization, carbon gas flux, and so on. You know, this is just a hypothesis to start with. And because much of the proportion of the root ends up quite deep in the soil, it's colder, it's less aerated, so less microbial activity. Conversely, you've got the fibrous root system on the other extreme. Obviously, there's all sorts of things in the middle, which I did take into account. But for the purpose of this talk, we'll just talk about these two. So you've got lots of root mass in the top. It's very warm, lots of air going on, lots of space for microbes to attach to the little tiny root pieces. And also, potentially, higher plasticity, because it's less high risk to just slough off a few that have made it to a bad point and maybe redirect a few roots into a high nutrient space or something like that, you know, or if it's droughted, you know, you can kind of shift. So that was my kind of working hypothesis at the beginning of the study. So I chose some functional traits that were very easily accessible on online databases. And I made a species list of chalk grassland 3A community, which is the National Vegetation Classification for this site. So this area of chalk grassland on Salisbury Plain. And so I went through all the literature, all these online databases, made my trait lists. And then based on these traits. And so you can kind of see, you know, how they would have different effects on the soil. And it's never been done on chalk before. I had a species list of something like 250 species. It was insane. But so it's never been done on chalk. It's been done on mesotrophic grassland, acid grassland, and so on. But this was a really interesting challenge because most chalk grassland species are very slow, very small. They take forever to establish. And I was a bit worried that they'd all be identical. You know, they'd all be tiny and rubbish and live for 50 years. And actually, quite a few of them did. But I did a cluster analysis, and what I came up with were 3 functional groups, which is actually what I asked for. This isn't me being disingenuous. I did ask for 3 functional groups, and on the top, these 3 are just an example of 3 of the species that we got in the first group. And so they're the ones with the thick taproot. So you can make this hypothesis that poor activity. And then the group 3, which is the other one I want you to focus on, has the shallow fibrous roots. So this is just a little picture of the soil on the site before and after. It did grow incredibly fast, actually, which is not what I expected, really. But you can see the substrate we were working with. So this is quite complicated, and all I really want you to take from this is that on the left you have where the red line is where the group is absent, and the little dots are where they're present. So you've got group 1, 2, and 3, so tap, tiny rosette, and fibrous. And basically, the processing rates on the left are all to the left of the line, which means that they're all pretty slow, which is what I hypothesized in the beginning. And then the ones in the middle, which I thought were going to be really tiny and maybe not have much influence, because they are tiny, they're all kind of on the middle line, so they're a bit naff and they're not really doing anything much. And then the ones on the right generally fall to the right. Now, this is just a snapshot in time. This is just July 2015. And so I'm going to go into more detail. This is a time series of 2015. And on the left, you've got where the group 1 is present, so mineralization at the top and extractable nitrate as a result on the bottom. And the thick line is where the group is present. So like the average of where the group is present, and the dotted is where it's absent. And you can see that where you've got the tap roots, it's lower than average, and where you've got the fibrous roots, it's above average consistently. So this is extractable nitrogen. And so of course with the fibrous roots, that's not very desirable in this system because it's going to be lost. And on a landscape scale, that could end up in the watercourse. You know, there's nitrate, and you end up with eutrophication and so on. So it's not very desirable. This is just functional group 3. This is respiration, so carbon mineralization and loss to the atmosphere on the left, and photosynthetic rate on the right. And you can see that these rates are much, much higher when you have the fibrous roots. But when you have the drought, which I'm going to talk about in a minute, you end up with much lower, you end up with a big drop in photosynthesis, which is where this red circle is. It's a bit of a problem. So just to walk you through then, this is where I did a bit of model averaging using the actual trait means, and this is FD-var is variation in the plot, so variation of the trait in the plot level. And so basically with the ecosystem respiration, you get highest respiration and carbon loss when you have a very uniform life length, which is a bit surprising given that they're all, like, for the most part, they're very long-lived. So I didn't expect that. And you get the same trend with photosynthesis. And photosynthesis, the more negative, the higher the photosynthesis. But basically, everything got thrown out. The species richness got thrown out of the model, and Shannon's richness, and except for respiration. And so with the photosynthesis, it's all about the root traits, which was quite unexpected. But it does add weight to my hypothesis that traits are more important than species richness. So we did a drought treatment, 45 days of drought, fully excluded, nothing at all, and roofed to account for artifacts. And basically, if you look at this picture, this is Plantago. Plantago is not meant to be fluffy. And so everything we were growing was fluffy, and so it was already adapted to drought. And so we have found virtually no effect. And so my resistance measures were pretty much nothing. So there's a bit of mineralization rate, and as you have deeper roots, or more variation in root depth, you have higher resistance to drought in these two key processes that I've put here in this graph. So in conclusion, I think these categorical root traits are very important because they're easily explainable to land managers and farmers and so on, and they're easily accessible. You can get them anywhere, and you can just look at your sward and you can have a sort of basic understanding of what's going on under the soil. And I think that's quite a useful message. I know it's not, enormously accurate for our sort of, exact processing rates and so on, but for a land manager, I think that something like this is actually very valuable. And longevity is a lot more important than a lot of studies give credit to, and that has implications for turnover and so on. So so I think things like this are very important. So I'd just like to thank everyone that helped, and my funding bodies, and I'm going to plug our meeting for next year for the plant soil ecosystem group. We're going for winter ecology, so please come to that.
Thanks, Ellen. That was really fascinating and great amount of work. Any questions for anyone? We've got one.
I just one with the plant traits. How applicable are they to something like calcareous grass? Are there any that are developed on those sort of conditions, or is it just a very much a general database, from plants in all sorts of habitats?
It was the species that you would expect. The species that I planted were the ones that I used. So I would hope that the database values would match, because I was going for sort of mostly categorical as well, sometimes never or always mycorrhizal, or because they're not going to suddenly become a tatic. So that's why I wanted it to be applicable across, if I doubted it, they're not going to suddenly change.
Yes, I'm doing this.
So yes, that's what I thought.
It's all right. We've rolled over to the next slot anyway. So Lars Gemfeldt is going to talk to us about biodiversity cannot directly affect levels of ecosystem multifunctionality. And that was a very smooth changeover.
Okay, thank you very much. So my name is Lars Gemfeldt, and I'm here to present some work that I'm doing together with my colleague Fabian Roger, who is, by the way, the next presenter in this session, so please stick around for that one. As you're probably all aware, scientists have long been fascinated about the consequences of biodiversity loss. And I think it's fair to say that we today know that, generally speaking, on average, biodiversity loss has detrimental effects on ecosystem functioning. One issue that I've been pondering quite a lot over the last decade or so is the importance of biodiversity for the simultaneous performance of multiple ecosystem functions, often referred to as multifunctionality. And since species differ in their contributions to different functions, you would propose perhaps that a high variety of species are needed to sustain all those functions. And for example, Emmett Duffy and colleagues, they found back in 2003 that a diverse assemblage of small marine grasses simultaneously maximized multiple functions in a seagrass system. And positive biodiversity multifunctionality relationships have later been shown for many systems, for many groups of organisms. And a recent meta-analysis from last year suggests that this relationship is quite general as well. And the concept of biodiversity and multifunctionality, I would say, has become somewhat of a bandwagon in ecology and conservation. There's more and more papers emerging each year. And the importance of biodiversity for multifunctionality is grounded on a verbal framework that makes intuitive sense, right? Species have trade-offs in their and how they allocate resources to growth and defense and reproduction, so no single one species can maximize all functions. So you would expect that you need more species, of course, for a multifunctional landscape. However, this intuitive knowledge is based on vague terminology that I think has painted a somewhat hazy picture of a causal relationship between biodiversity and multifunctionality. You see an example here on the screen, a paper from 2008, in which the authors of the paper talk about something complementarity across multiple functions, whatever that is. And we argue now that it is time that scientists abandon the idea that biodiversity directly and causally affect multifunctionality. I think many people here today will find that this is probably the most trivial talk you have attended. I think I hope it's not, it's very trivial and basic, but I think it's worthwhile going through everything anyway. So biodiversity can be proposed to be important for multifunctionality in two major ways. The first one is that a variety of species provides for a variety of functions, right? And so if certain functions are unique to certain species, you would need all those species for a functional system. And to take a very simple example is that you lose a herbivore and a predator species from a system with one plant species. I would say that this is very trivial. And most importantly, this is not what the studies on biodiversity and multifunctionality have been concerned with. Pretty much all the papers on the subject have been concerned with the second way that biodiversity affects multifunctionality, and that is that biodiversity affects the level and the magnitude of multifunctionality. That is what the papers are about. And most of the papers then, all of these papers, they study a defined set of functions shared by all species, mostly for one trophic level. So you go from high diversity to low diversity. You examine, say, productivity, nitrogen uptake, weed suppression, and you examine the consequences of biodiversity change. So we now going to demonstrate that under the null assumption, a null model, which is the common null model in biodiversity functioning studies, where we assume no niches and equal fitness across species, that biodiversity does not directly drive multifunctionality. And we also show that one of the most prominent claims for the value of biodiversity when it comes to multifunctionality is that as we start consider, we move beyond considering just one function, we consider more and more and more functions, biodiversity somehow becomes more important. So take a very simple example, 3 species, X, Y, Z, 3 functions, F1, F2, F3, the mixture of the species. We have here standardized, in all our models, we standardize the functions to range between 0 and 1, just for simplicity. You see here that the best species varies across the 3 functions here, And for the mix, and for all monocultures and the mixture, the average in the blue bars stays the same. If we expand this, and let's now model 15 species and 9 functions, it is then trivial, under the same assumptions, that there will be no relationship between richness and any of the individual functions, right? Each function can be on slightly higher or lower level, but there's no, the slope is flat. And then if we look at the average value of these functions, which is a very common metric of multifunctionality in the literature, then it's also trivial. I think trivial and multifunctionality are words I'm using a lot, but I think I need to. There's, of course, no relationship either. You have the average functional value there on the y-axis, and again, richness on the x-axis. Let's then take another commonly used metric, often recommended in the literature, which is to look at how many functions are sustained above a certain threshold. I'm now returning to the simple example with the 3 species and the 3 functions, and let's look at the 40% and the 60% threshold of the maximum value, functional values. Then another picture emerges. For the 40% threshold, 2 functions on average are above the threshold for the monocultures and all functions for the mixture. But at the 60% threshold, so there's a positive biodiversity effect there, right? For the 60% threshold, you have a negative relationship. 0 functions are provided for the mixture. And let's return now again to our modeling exercise with the 15 species and 9 functions, and plot slightly more thresholds here. So we have the number of functions above the threshold on the y-axis, and richness on the x. And we see that there will be a positive slope for all thresholds below 50% and negative above. Remember, this is still under the assumption of no niches and equal fitness for species. And please now remember the central panel there, where the slope is flat, the 50% threshold, and because now we're going to look at the change in the number of functions per addition of 1 species. Now we're plotting the slopes from the previous slide. Now we're plotting the slopes here, but we do it for each percentage from 1, 2, 3, 4, up, all the way up to 99%. And again, this is just another way of visualizing things. You see here that the slopes, which we plot now, are positive below 50%. 50% is at the point P here, and it's negative above. And now, remember that point P here. That is where the relationship turns from positive to negative, because that's what we're going to plot on the next slide here. And now, because one of the most prominent claims I said for the value of biodiversity for multifunctionality is that as you start considering more functions, then we're going to see the value of biodiversity. But if we plot, you see the point P up on the inset there. If we plot that on the point P on the y-axis as a function of the number of functions, so to speak, we now increase number of functions. It's a slightly different model, but same assumptions. We could go up to 1,000 to 1,000,000 functions here. It doesn't matter. The relationship is flat. It doesn't change. It doesn't matter how many functions we consider. And again, trivial, I would say. Biodiversity can only affect multifunctionality via its effect on single functions then. So say that we would instead model, we would take our model, and then now we include what is a classic biodiversity effect in the literature. We include a complementarity effect for 2 of the functions, 2 out of 9. So complementarity is something like niche partitioning or positive interactions and so forth. Those 2 individually positive relationships will then transfer to an overall picture that you see here with a positive relationship between species richness and now this average functional value metric, which is like a slope you commonly see in the literature. So this simple model and these simple graphs that I walked you through, this is what we want to communicate. Like, given no individual diversity effect for any of the functions, it doesn't matter if you consider a million functions. The relationship between biodiversity and multifunctionality does not count. And however, we do want to emphasize that we don't tell people to stop looking at multifunctionality here. Multifunctionality is very important to study if we want to understand how functions, not maybe understand, but to summarize multiple aspects of functioning and how that responds to global change or multiple stressors or biodiversity loss and so forth. And multifunctionality metrics can be very useful in understanding the collective behavior and understanding trade-offs among functions as well. But we want to emphasize that it's time that we understand that there is no mechanistic direct link, no causal link directly from biodiversity to multifunctionality. And if we continue to focus on that, continue writing that in our papers, that would only lead future research and conservation efforts astray, I think. It's almost like, for me, this finally realizing that the multifunctionality emperor is naked. So that's, and with that, I'm happy to take any questions. And now thank you very much for your attention.
Terrific, Lars. That was awesome. Very controversial, thought-provoking. So any questions from people?
Very nice talk, Lars. I guess when you have the positive biodiversity effect for each of the functions, but it differs with respect to how it works, then you would have this positive effect between diversity and multifunctionality. I don't know if you put this.
So you have different effects for different functions, or?
Yes. I mean, your simple model has basically the individual species effect, so there's no biodiversity.
Yeah.
Now, if you have positive interactions, but the type of interactions depending on the functions and also what species basically are involved in this positive effect of.
Yeah.
Then, if you increase species richness, you will have all these positive biodiversity effects, and then you would.
Exactly, but that's the point. Then you have you go into like something like how plants partition their resources, so a mixture of plants is more productive than a single, and then the mixture produces more biomass, and hence it might take up more nitrogen. It might provide more habitat for insects and so forth. So you have different functions driving each other. I think people need to start thinking think better about and more thoroughly about what functions they study and why, because that is when you get these multifunctionality effects, right? You have one function affecting another function, but it's nothing magical happen between biodiversity and multifunctionality per se, which all the papers claim, right? Oh, biodiversity, consider more functions, now it's more important. That we need to quit. But please study multiple functions by all means, I think that's good.
Any other questions?
It was so trivial.
Mm-hmm.
You calculate multifunctionality by taking the arithmetic mean, I
Yeah.
think, the different functions. But one could also consider some geometric mean.
Yeah, sure.
And then in your monocultures, many functions could be absent, which decreases multifunctionality compared to the biodiverse culture, where at least all of them are present to a certain extent. Would that change the implications of the model?
No, I don't think so. No, we tried many different models for this. I need to think about it for a while, but no, I don't think it would. We tried different metrics and we simulated. Fabian here he's created a little. If we get this published, hopefully, there's going to be an app provided. We can play around with these different things, different values of complementarity and levels of the functioning and different means. We can incorporate that. I'm pretty sure it's not going to matter.
Great, thank you. We might move on.
Yeah.
So it sounds like we're going to have a nice transition here between the two talks. So our next speaker is Fabio and Roger. I probably mispronounced your surname, so apologies.
That's fine.
And we're going to listen to habitat diversity and multifunctionality.
So, thank you for sticking along and staying for my talk too. Just before I start, I would like to say that the talk I presented today is Christian's postdoc work. He presented yesterday, Habitat Diversity Strengthens Primary Productivity. So if you haven't seen that talk yet, go back and see that first. And yeah, Christian was a bit nervous this morning. He said, like, I don't know why I feel nervous. It's you presenting. And I said, yeah, Christian, that's because I'm presenting your work. So I'm not the only one to blame in either case. What I would like to talk about is habitat diversity and ecosystem functioning, or as we write it, habitat diversity and multifunctionality, because to make that point again, we still think multifunctionality is a valid summary metric if you need one single response variable and cannot deal with multiple response variables. So, why talking about habitat diversity? Well, we know that species diversity can drive ecosystem functioning. I think most of us agree that in general, productivity, for example, increases with species richness. But if you go and look on the convention on biological diversity and how it defines diversity, you will read that biological diversity is defined as the variability among living organisms from all sources, including diversity between species, within species, but also ecosystems, or as we put it, habitats. And the BEF literature, BEF being biodiversity, ecosystem functioning literature, has so far solely focused on the diversity between species and a bit within species genetic diversity. But there hasn't been studies of the diversity effects of habitats. So we are asking whether this level of organization can also be important for landscape functionality, especially in the light of habitat homogenization, where we're not only losing species, but we're also losing habitat complexity and habitat diversity. I think we should start considering that level of organization too. This is not to say that there hasn't been studies looking at habitat-habitat interactions. So there's a study of Schindler and Charrel here, where they look at different habitats in a lake and describe how they interact, how they exchange materials and energy, and also the coupling in general of riparian forests and lake ecosystems or river ecosystems. I think it's a well-known example of habitat-habitat interactions. If there are marine biologists among us, also the pelagic benthos coupling observed in marine systems, where you have the euphotic zone in the pelagic surface habitat that exports energy to the benthos, where nutrient remineralization happens in a very simplified way here that provides nutrient again to the pelagic zone. So habitat-habitat interactions have been studied and recognized, but habitat-habitat interactions, we'd argue, is equivalent to species-species interactions. And as species-species interaction in the end underlie species diversity, ecosystem functioning relationships we find, we also think that habitat-habitat interactions will underlie habitat diversity effects. But they haven't been studied in a diversity ecosystem functioning framework. So we work in Sweden, in Gothenburg, and the West Coast, we get these lovely bays that have a variety of different sediments, or I'd like to call them the 50 shades of mud. And for the bacterial communities that drive the functions we looked at, which is gross primary production, oxygen flux, or denitrification and nitrogen fixation, which again, as you will see later, we summarized in a multifunctionality metric using the standardized mean of those functions. For these organisms, those different mud types present very different habitats, very different biogeochemical habitats. And what you see here, you have sand on the upper, you have rupia meadow. On your right, you have a more silty, more anoxic habitat. You have also cyanobacterial mats and zoster meadows, which we didn't include in this study. So what we did is that, and some of you might recognize some of the slides that I borrowed from Christian, but we assembled those cores. We took cores from all those separate habitats and assembled them in artificial landscapes with varying habitat diversity. And so these habitats could interact via the common water column. And on the right, you see the layout of the experiment, which will be very familiar for all of you that are familiar with biodiversity ecosystem functioning studies, where you have every habitat in monoculture and then in combination of 2, 3, or 4 habitats. And so I'm gonna straight jump to the first result, which is the net diversity effect, only looking at the highest habitat diversity level, where you see that, so on the left, you have the expected functioning of the highest habitat diversity level by simply looking at how do the single habitats perform, and then what do we expect to perform on average if we put them together. And on the right, you have the observed diversity effect, and we see that we have actually quite a strong diversity effect. But before we go further, I would like to take some time and talk about which kind of diversity effects we can expect if we talk about habitat diversity as opposed to species diversity. And if you look here on the right, species diversity, one effect we are probably most of us are familiar with is the sampling effect in combination with the selection effect, where a high-performing species is selected for increase in abundance over time and therefore drives the diversity effect. This cannot happen in our experimental settings. The cores don't change in surface area, and the cores are also not selected for. Same goes for niche partitioning, as we heard again from Lars in the previous talk, an important diversity effect, which allow basically that a habitat can harbor a different amount of species or a different number of individuals depending on how well they utilize the total niche space. But there is no niche space for habitats. There is no habitats for habitats. The total habitat cover did not change over the course of our experiment. So we are left with positive interactions over the water column, exchanges of flux of energy and material. And so with that in mind, I'd like to point out that we did observe a habitat diversity effect of 66%. So there's one thing that is special with habitat diversity, is that if you manipulate habitat diversity, you're also manipulating species diversity. Because if you have species turnover between the habitats, the gamma diversity will be higher if you have more habitats. And species diversity, we know that it can drive multifunctionality. So we had to disentangle this effect. And to do that, most processes are driven by bacteria in these systems. So we looked at bacterial diversity, we sequenced it, and calculated bacterial gamma diversity in those big cores, and tested the effects in a structural equation modeling framework. And what I didn't tell you either so far is that we repeated that 3 times in summer, spring, and autumn. And I conveniently leave out spring, where we didn't find any effect. But for summer and autumn, we find at least statistical evidence that both processes operate, so that habitat diversity can directly influence multifunctionality, as we found in the summer, or indirectly influence ecosystem functioning or multifunctionality via a greater gamma diversity. And on that note, I saw that I rushed through my talk. I would like to end and tell your friends and colleagues that diversity in ecosystem functioning research should give a thought about that level of organization, of diversity, and whether it matters or not for landscape ecosystem functioning. We argue that habitat diversity is important, and we think we have provided an example in which it was shown to be important for landscape multifunctionality, and the effects can be direct or mediated by species diversity. And with that, I'd like to point out that the study was recently accepted. It's published as a preprint on PRJ. You can find it there, although some of the analyses are a bit outdated, and the final version is accepted in Science Advances. So if you look for it, you can read all the details that I didn't have time to go into. Thank you for your attention.
Thanks, Fabian. That was great. So we've got plenty of time for questions. So have we got any?
Yeah? Multifunctionality?
Or maybe I missed it. Yeah.
Okay, yeah, so sorry. So we measured 4 functions. I'm going to show you this slide here, maybe. So we have denitrification, nitrogen fixation, din uptake, might have actually confused them, and gross primary production. And all those functions were standardized between 0 and 1. And then we took the average functioning approach from Mestre et al., where you took the average of the standardized function value as metric of multifunctionality. Yeah?
Well, I was wondering, is there any connection? There's a lot of knowledge about metapopulation theories, etc. Or did you try to take one part out of all that research to see if habitats by their own have an impact? Or do you incorporate all those population dynamics in?
So I'm not quite sure I follow, but so what these habitats, we have the habitat functioning on their own because we have them in single treatments, right? And if you talk about metapopulation, I guess you're talking about exchange of species between the cores. This could have happened. They were connected over the common water column. Although most processes happen in the sediment. I'm probably not answering your question, but what I think is important is that we don't need to have necessary species turnover to get complementarity between the species in the different habitats, because I think that a species assemblage can perform context dependent. So the same assemblage in 2 different habitats might have different functions, especially those microbial functions, depending on the oxygen level or the nitrogen levels. That will change which functions the community performs more. And therefore, the same community, at least as I picture it, could be complementarity with itself if the same community is present in 2 different contexts.
Well, and does your, because you do experiment with multiple habitats, is that the same as heterogeneity?
So this is a good question, and what we used here, I didn't have time to go into that, is we used the continuous metric of habitat diversity. So what it presented, the net diversity effect, that's just the 1 versus 4. But in the study and for the SEM, we looked at the continuous metric of habitat diversity, where we quantified or we measured what we could think of in those habitats. Christian knows the details, and then plotted the habitats on as we basically calculate distance matrix between each small core. So even also including the cores of the same habitats, and then took the average distance between those habitats as a metric of habitat diversity. Yeah, so this goes in the direction of heterogeneity. I think heterogeneity is mostly studied in the sense that it allows for more niches and more total species diversity, and not so much for whether in a heterogeneous landscape you would have microhabitats that could interact with each other. But it's certainly something that has to be defined and is related to that research.
Great, thanks.
Thank you.
We'll have to leave it there.
Yeah.
So thanks again. And her talk is entitled Diversity Enhances Performance of Methane Oxidizing Bacteria. Thanks very much. Where do I find the full screen mode? This that you wanted? Will I get you that? Should do. Yeah, you got it.
Okay, thanks. Hello everyone, welcome to my talk. I'm going to present an experiment that I did as part of my thesis on biodiversity ecosystem function relationship in methanotrophic bacteria. Research over the past 20 or so years has shown that biodiversity enhances the ecosystem functions of such as plant biomass production or resource uptake or nutrient cycling. Here in this example, you can see that plant biomass increases with species richness. But the research so far has focused mainly on above-ground organisms, particularly plants, and much less has been done on microorganisms. Here you can see a comparison between the number of studies that have manipulated the biodiversity of plants and microorganisms. But microorganisms are very important for many ecosystem functions, and so far we don't know much about what happens if we lose diversity of microbes. This is true for general microbial systems, but also particularly for soil systems, and soil communities are extremely diverse, and they are fundamental for many biogeochemical processes. Such as in this example, you can see that bacteria are involved in many steps of the nitrogen cycle. And climate change and land use intensification threaten both the abundance and the diversity of microorganisms. So far, there have only been a few studies that have looked at the biodiversity ecosystem function relationships in microbes. And the results of these studies have been rather inconclusive. And some studies have found a relationship, such as the bell here, but other studies haven't found any. And that may be in part because they looked at general functions such as respiration or decomposition, which are carried out by a large number of microorganisms and which have high redundancy. Microbes that carry out more specialized function have been studied much less, and as they are less diverse and therefore probably less redundant, they may be more affected by diversity loss. So in my thesis will look at the methanotrophic bacteria, which carry out a very specific function, that is, they oxidize methane to carbon dioxide, and they are a phylogenetically very narrow group. They live in very diverse environments, such as rice paddies, soils, wetlands, and landfills, and they are autotrophs, and they use methane as their only source of carbon and energy. They are significant for the global methane budget, and as methane is the second most important anthropogenic climate gas, they also play an important role for climate warming. Methane is about 30 times more active per molecule than CO2, and it accounts for about 20% of the current global warming. So methanotrophs work on this system in two ways. They are, on one hand, a direct sink for atmospheric methane. They account for about 5% of the global methane sink. But much more importantly, they live in the same environments as the methane-producing archaea, and they oxidize up to 90% or even more of the produced methane before it enters the atmosphere. So they are key for understanding the regulation of methane emission. And as they're also not highly diverse in nature, they may be particularly affected by diversity loss. So what we wanted to look at in this experiment is whether the biodiversity of methanotrophic bacteria influences the ecosystem function methane oxidation, and also the associated production of microbial biomass. To do that, we mixed communities from 10 pure methanotrophic cultures into 5 different diversity levels. So monoculture, 2, 4, 6, and 10 species mixtures. For each diversity level, we had 10 different compositions, except for the 10 species mixtures. And we made sure that each species occurs in the same number of communities per diversity level, so that we didn't have a confounding influence of composition and diversity. And these communities were grown together in microcosms that consisted of sterilized soil that were put in these tubes. And they were incubated in these jars under an atmosphere that contained 1% methane. Of each composition, we had 7 replicates that we removed at different time points so that we could look at how the communities develop over time. From 3 of these time points, we extracted DNA, and they were the beginning, the end, and the point in the middle of the experiment. And we sequenced the communities and then calculated from this the number of strains that were present or that we could find again from the ones that we put in, and the relative abundance. And from that, we used some as our diversity measure. So we have applied richness. That is the number of species that we put in, so our original diversity levels. And then we have realized richness, which is the number of species still present at the end of the experiment, and the effective species richness, which incorporates this relative abundance of these species. Here, just to illustrate, we have the number of species we added, plotted against the number of species that we still found at the end of the experiment. And as you can see, we lost some species, especially in the high diversity levels, and there was also some strange things happening here. But we still had a diversity gradient at the end, and the evenness also went down quite dramatically. To measure methane oxidation, we took gas samples from each jar over the course of 2 days, and measured the methane oxidation, and then after that we replenished the methane in the jars again. And then from the change in the methane concentration, we calculated this oxidation rate constant K based on the first order kinetics over each of these 2-day periods. Here you can see the methane oxidation rate over the time of the whole experiment aggregated by the applied richness. You can see the methane oxidation rate went up quite strongly in the beginning and then decreased again towards the end. And also that the difference between the diversity levels is highest here, where the oxidation rate is also highest. Here you can see the mean oxidation rate over the whole period plotted against applied richness and realized richness. And both show that there is a significantly higher methane oxidation rate in higher diverse communities. The effective species richness was not significant, but we often find this in our studies, and we interpreted that the abundance is not so important, so that even like rare species are important for the ecosystem function. We also did qPCR on the same samples that we used for DNA extraction to measure biomass, and we used the gene copy number of a methanotrophic specific gene as a proxy for biomass. So here is the biomass at the beginning of the experiment, and then at the end. You can see that the biomass increased in all diversity levels, but considerably more in more diverse communities. So by the end, we have this nice biomass diversity gradient. We also observed an over-yielding effect for both methane oxidation and biomass, so that the observed methane oxidation rate for almost all mixtures is higher than the predicted, which is the mean of the monocultures that were in these mixtures. And for biomass, we see something similar. So to summarize the results, higher methanotroph diversity increases methane oxidation and microbial biomass. We also tried to relate this to phylogenetic diversity or functional diversity based on the phylogenetic distance of the strains in the mixtures or their differences in traits, but we didn't find any clear results there. So in conclusion, I can say that diversity increases the performance of methanotrophic bacteria and that these strains show a positive interaction. And to go back to microbial biodiversity ecosystem relationships, at least in my system, I could find the same results that have been found in plants. And we can assume, therefore, that at least in some microorganisms, they show the similar patterns in different scales and in different organisms. But we need more studies that look at biodiversity ecosystem functioning relationships also in other microorganisms, so that we can make more general predictions about the effect of biodiversity loss on their ecosystem function. And also specifically for methanotrophic species, it is possible that the diversity is also important for maintaining methane oxidation in natural communities. So it is crucial that we maintain their diversity for mediating methane emission and to combat climate change. With that, I would like to thank my supervisors and co-authors, my helpers, and thank you all for your attention.
Thanks, Elvira. That was really good. So we've got 3 minutes for questions, which is lovely. So nice time for discussion. So has anyone got one? Thanks.
Nice piece of work. I was just wondering if the photographs that you selected, they come from the same type of environment, from the same ecosystem?
No, they are quite difficult to cultivate. So these 10 were just what I could get my hands on. So. Yes?
Could you comment on how this diversity that you have compares in any way to the natural diversity of methanotrophs? What you would find in a metagenomic sequencing of soil? Or?
Well, not quantitatively, but usually you don't find a lot of specific OTUs when you study the soil directly. So, yeah, I mean, you find a lot more than have been cultivated, but you can't really say. Yeah, it's not a lot in nature either. So if you look at the soil from a wetland or something.
Thanks.
Yes?
Thanks. Yeah.
In contrast to experience with plants, with your bacteria, you know exactly the trait and its activity per each species. So I wonder, because you had them all in monoculture, also you know the specific activity of each bacterial species. Now, does the presence or absence of one species in your mixtures explain your results?
To a degree, there is one strain that is significant if I put it in my model. So this is Methylococcus Methylocytis echonides, but this one isn't the one that performs highest in monoculture, so there is still an interaction.
Do you see transgressive overyielding? So the best monoculture still performing worse than the highly species rich community?
Not really. So maybe we can go back. Here you can see, so this is the highest monocultures and this is the highest mixture, so it's not really transgressive.
So that yellow point is also present in the red.
I can't say for sure, but like these 2 points here, the highest monocultures, only one of them is present in these 2, for example. So it's not just these 2 that explain everything.
And I've got a quick question. Would you expect interactions with bacteria and other organisms, other different types of organisms?
Yeah, definitely. Yeah. In my second experiment, we did a dilution series so that we could also have these other microbes.
Okay.
Experiment. Yeah.
Right. And that perfectly takes us to time, so we'll switch over to the next speaker. Thanks very much, Elvira. So our next speaker, and I'm really going to get this wrong, so I apologize in advance, is Zhao Jingyuan. Is that, maybe you can say your name?
做强援.
Yeah, it's exactly the same as what I said, wasn't it? And he's going to talk to us about a disturbance-induced increase in tree species diversity does not enhance carbon storage in temperate forest.
Hello, everyone. I'm Zhuo Qiang. Today, I want to talk about the diversity effects on carbon storage in temperate forests. We know by diversity and ecosystem functioning, relationship have been a subject of considerable interest in the past decade due to the accelerating species loss rate. And recently, a meta-analysis of the diversity multiplication studied in forests suggests the mixed species stand was more productive than the monoculture. However, whether the result from the controlled experiment can be extended to the real ecosystems with open questions. And the result from the observation studied in forests have shown mixed evidence for relationships, positive and negative or neutral. And there are two mechanisms in BF relationships. One is the niche complementary hypothesis, suggests the improved ecosystem process due to the niche partitioning or facilitation. Other is the selection effect hypothesis, suggests the high diversity means a high probability of including one or few dominant species. This hypothesis both simultaneously underlies the net effect of diversity effect on the ecosystem function. And another hypothesis is green mass radio hypothesis. This hypothesis suggests the ecosystem function is mainly determined by the function trees of dominant species, and not have much relationship with the diversity. And the effects can be estimated by the function identity. For example, the community weight mean of certain function traits. And an interesting question in the BF relationship is which diversity measure is a better predictor. And the recent results suggest the functional and phylogenetic diversity was a better predictor than species richness. But some results suggest not. And one of my recent studies showed that the multiple metrics of diversity have different effects on temperate forest over succession. So we can't say which diversity is more better or not. And another driver of the BF may be abiotic variables in nature forests. Some studies show that if we remove the effect of the abiotic variables, the relationship may be changed. And the disturbance in forests, such as logging, is also a driver of the BF relationship. And the effect of disturbance mainly depends on the frequency and the type. So there is a great uncertainty of the magnitude and the direction of the BF. And we also ignore the mass radio effects, for example, the function tree role in the BF studies. And the recent reference suggests that the function trees have great power than the diversity. So my question in this study is, I want to test which measures of diversity and the function identity are the best process of complementarity and the mass radio hypothesis, and quantify how the relative importance of diversity and the functional identity change in disturbed and undisturbed forest. And my study site is located in the broad-leaved currant pine mixed forest in northeast China. It's the typical vegetation type of in this area, but now is nearly disappeared due to the human activity. And only occurred in the residual fragments. So we established the standard perimeter plot in the residual fragment of the forest. 7 was distributed. This red cycle is the Changbai Mountain Nature Reserve area, and 5 in this reserve, and 7 outside the reserve. And the 7 was suffered from occasional logging for timber, and now it has stopped the logging in 1999. And this table tells us the basic information of the plot. In the disturbed plot, they rarely have large trees, and but have high species richness. And the undisturbed forest was including much more large trees, but less species richness. We also calculate the different diversity index, including taxonomic diversity, phylogeny diversity, and function diversity, and also functional identity, and also topography factors. We based our following analysis based on 10 by 10 project, because in our prior study showed that the species interaction mainly occurred at this scale. And we first test the spatial correlation in our carbon storage. We construct the GLS models with and without spherical autocorrelation structure, and the results indicate that the models without the spherical autocorrelation structure will always have the lowest AIC value. So we didn't consider the spatial autocorrelation in the following analysis. And we also, because we have calculated many variables, so we use step-wide method to select the variables, and then construct a series univariate and multivariate linear mixed model to evaluate the combined effect of the above the three variables, like this, univariate and multivariate. Since there were strong correlation between some variables, so we avoiding including more. And we also do the model averaging approach and the variation partitioning method depose the variation in the soil and the carbon storage into into different part of the explained variables. This is our result. The disturbed forest had a higher diversity in terms of maximum species Shannon-Wiener index and phylogeny diversity, but have low functional evenness and dispersion diversity. This model comparison result. The best model include the CWM of maximum height and evenness and the Shannon-Wiener index. And in disturbed plot, in disturbed plot and the undisturbed plot have the similar result. The variation partitioning results show the function identity have great importance than the diversity, regardless of in disturbed forest and undisturbed forest. However, in undisturbed forest have more variation explained by diversity and function identity. This model averaging result tell us which variables were more important. We can find the Shannon-Wiener index was more important, and the maximum height is a measure of functional diversity. Is the most important factor that determine the carbon storage. And our biotic variables was not significant influence because our approach is very gentle. So there are several previous studies that have found poor or no support for nature complementary hypothesis on diversity. And some emphasize the mass residue hypothesis, and our results prove that these two effects simultaneously enhance the ecosystem, the carbon storage. However, some studies suggest diversity was as important as functional identity in explaining carbon storage, but our results showed that the diversity was very weak. The role was very weak because maybe the complementary effect may be more important in water-limited forest or in fertile soil forest. As other explanation, maybe we use less function trees. And our result found that the heat is more important than wood density, because one important reason is the dominant species is pine coring, is conifer trees. Have high maximum heat but low wood density in our plot. And in disturbance forest, in undisturbed forest, these two effects both increased, suggests that the both effects will increase as the forest incurring from disturbance over time. So that's our conclusion. We found that these two effects both affect the carbon storage, and the mass residue effect was more powerful. And the disturbance induced in species regions didn't enhance carbon storage because it results in more functional similar species. Our result implies that both effects increase with forest recovering, and if we want to maximization the forest carbon storage, we require protection of species with the trees, also to raise the maximum heat and this area. Thank you.
Thanks very much. That was a great study. Impressive. Any questions? So I've got one. So I was just wondering, did you just look at the woody species, so the trees, or did you look at understory species as well?
Yes.
Just the trees?
Just trees.
Yeah. Do you think that understory would contribute much to carbon storage, or not really? Not in comparison to the trees.
You mean the understory species?
Yeah. Is there much of an understory?
Yes, but the understory species not very much.
Okay.
Yeah.
Any other questions? Or we might switch over.
Yes,
Thanks
okay.
very much. That was really good.
Where are the talks saved?
Our next speaker in this session is Biomass, and his talk is entitled "The Impact of Disturbance on Biodiversity and Ecosystem Functioning in Multitrophic Communities."
Thank you very much. As noted before, now we are coming to multitrophic biodiversity and how disturbances influence biodiversity and subsequent function in these multitrophic communities. I have to warn you that will take you a little bit into the world of food web modeling. As already stated before in the talk by Lars, most biodiversity and ecosystem functioning studies are focused on plant communities, on the diversity of plant communities and their ecosystem functioning. As an example, in a meta-study from Balvanera et al., they found that over 200 studies at that time focused on that relationship. We measured the diversity of plants or manipulated the diversity of plants, and they measured these studies some function, also most often on the level of diversity of plants, or on the plant trophic level. And just 5 out of the studies they found investigated the effects of biodiversity on multitrophic scales and the effect of or the functions on these multitrophic communities. And on the other hand, researchers, scientists that are focusing on studying multitrophic systems, for instance, food web studies, are most often interested in how the diversity and the complexity of these multitrophic communities, for instance, food webs, affect stability of these systems. That can be, of course, also, for instance, persistence, meaning the amount of species survive. So how does biodiversity influence biodiversity in a multitrophic setting? But can be also, of course, other measures of stability, but I will not go into detail on that. So the question is, how should we or how can we combine these two different frameworks? So on the one hand side, we have the biodiversity ecosystem functioning research area, and on the other side, we have food web research, multitrophic biodiversity questions related to stability. So one possibility is we take the framework of food webs, what we see here on the top, and put it into the biodiversity ecosystem functioning framework. So a food web comprises normally basal species, plant community, and then also higher trophic level, here denoted as nodes, that feed on the lower trophic levels, here denoted as lines. And here we have a guild of herbivores, and we have different level of carnivores. And with increasing species richness, so in that case, these comic of food webs, we increase the richness of the animal community. We have also a more complex community of animals. I guess there's even more studies try to combine these fields. That is just an example here. So in that study, what was also a modeling study, we could show that with increasing species richness, we have an increase of the total biomass of the whole animal community with increasing species richness here on the x-axis, whereas the total biomass of the plant community stays rather constant with a slight decrease with increasing animal species richness. So we turned the whole thing a little bit around. But we are also able with that framework to analyze and investigate subsequent and underlying functions. For instance, here just as an example, of the ecosystem respiration of animals, here denoted as total metabolism of the animal community, increases independence of the increase of the biomass density of the animal community that was caused by the increase of biodiversity of the animal community. But all these things have assumptions. So, for instance, biodiversity research often tries in their experiments to get a constant biodiversity. So they maintain the experiments, the large-scale experiments are also in the lab, that the communities can adapt over time to that biodiversity level, and then the function is measured. On the other hand, multitrophic projects, especially modeling in the modeling world, I'm more used to try to keep the environment where the food web, the multitrophic community is embedded in, constant. So no fluctuations of temperature, seasonality are often ignored. The same counts for other disturbances from the environment. So, but these things are not stable. So biodiversity is changing through time in natural environments. So we have extinctions on the local area, we have invasions and new and metapopulation dynamics, for instance. And we also have seasonality, we have disturbances over time, for instance, floodings or also intertidal changes. So everything that disturbs somehow this local biodiversity of a multitrophic community. So in that study, we want to exactly investigate how that non-stable but dynamic systems in terms of changing of biodiversity and changes in the environment due to disturbances affect the species richness of a multitrophic community and also the subsequent functions, here namely the productivity and the ecosystem respiration of the whole local community. Our toy was a food web, in that case a model food web, and here's a comic of that. As noted before, we have basal species here in red, and all are interconnected by feeding interactions to the higher trophic levels. So here in orange, the herbivores, and to the higher trophic level, to the carnivores, omnivores, that gets even more complex in terms of feeding interactions. And this model food web is not a standalone, an isolated food web. It is embedded in a regional set of food webs, or in our case, a regional species pool. So that means we have here a patch in the food web, and all the species in the food web are living in there, and around in the region there are more and more species surrounding that. So what happens now, or in our imagination, what could happen, for instance, if you have a flooding, some of the species can go locally extinct because they get killed or they get just flooded away. In the same vein, in the same process, it could be that now they have here an open niche space due to that disturbance, and that can be refilled from species or individuals from species of the regional species pool that might be flooded into the local patch at the same time due to the disturbance. Our framework or our instrument is modeling, so we build up a mathematical model, mix that with a trophic food web model, put that into the computer, shake it a little bit, and what we get is time series, how the biomass densities, for instance, in gram per square meters, but also individuals per square meters, whatever you want to calculate, change over time. And we stop that from time to time, so, and put in a disturbance and kicked out a little bit of the biomass of the species randomly. That is an example time series here, how that looked like for a 3-species food chain, a basal species in blue, intermediate species in red, in blue, basal in green, blue, red at the top. And here we have all that kind of disturbances here, denoted by that little lightning. And we see that a strong disturbance decreases the population density, in that case of the intermediate consumer, dramatically, followed by the top predator that now also has a strong disturbance and not enough to feed anymore and goes extinct afterwards. Over the next period of time, or that time series here, the predator tries to re-invade the system, but as its prey, the intermediate consumer, is not abundant enough, it is not able to re-invade the system just if the prey is high enough, and by chance it gets flushed into the system again, it is able to again sustain in that patch. What we did, we plowed that a little bit up to the food web context, and we didn't change the richness of the local food web, but we changed the richness of the regional species pool. We changed the strength of the disturbances, so how much biomass is taken out randomly from the different populations, and the number of disturbances over time. And we investigated how that affects the local food web structure and the change in biomasses, so how much biomass, standing stock we have in the system, and also ecosystem functioning, here focused on the primary productivity and the ecosystem respiration. What we found is that the mean local, not the regional, because that is a constant, or it's manipulated. First of all, the regional species richness didn't affect most of the things. So we focus here on disturbances and the number of disturbances and the disturbance strength that mainly have driven the results. In the upper row, you see that the mean local biodiversity of the food web increases with number of disturbances, but decreases with disturbance strength. So the more often a disturbance happens into the system, the more likely a new species gets flushed into the system. At the same time, disturbances, both the number and the strength, decreases the biomass densities because they put out biomass out of the system, but put some biomass of new species into the system. Ecosystem functionings that are underlying is productivity on the upper row and the ecosystem respiration. And we see that all functions here, both primary productivity and ecosystem respiration, both decline with increasing number of disturbances, whereas, and also they decline with disturbance strength. But we can now also calculate the ratio or the difference between these two things to come to a measure of net system productivity, something like being more carbon uptake or carbon release of the whole local ecosystem. And here, due to mismatches in the rates or the reaction of the rates to the disturbances, we found that the number of disturbances, with increasing disturbances, the net system productivity increases. So more carbon is bound relative to released in the systems, but the strength of disturbances decreases the whole thing. So let us come to the conclusions. So for me, I see that multitrophic models are able to provide a better understanding how biodiversity out in nature can be seen and how disturbances and also dynamics lead to different ecosystem biodiversity function, biodiversity and ecosystem function patterns. And disturbances influence biodiversity and their underlying functions. And in the extreme case, so we still have to figure that out, it might turn CO2 sources to CO2 sinks. So we still have to check that. And with that, I thank you for listening, all my colleagues and co-workers from the BEFMATE consortium here, and also my wonderful working group, EPO-NetLab at the IDIF in Jena, Halle, Leipzig, all the founders, and again, you for your attention. Thank you very much.
Another great talk. I chose well with this session. Thanks, Bjorn. Has anyone got any questions? Thanks up the back.
Thanks, great talk.
Thanks.
Maybe I just missed it, but what characterizes your species? And the follow-up question would then be, what defines biomass and respiration?
So it's an allometric model approach, so it's a differential equation model system, and all species are across a range of body masses, so the larger eating the smaller, and using then allometric relationships of metabolic rates, feeding rates, and so on, allows us to measure also the community level of metabolic rates, but explaining the whole model framework would have taken too long.
Thanks.
Another quick one? I've got a super quick one. So with your strength of disturbances and your frequency of disturbances, it seemed that the biomass effect of them was similar. So do you know why strength is having a much bigger impact, therefore? Or why it's having the opposite, causing the opposite trend to...
With the biomass, or with the biodiversity?
I think, so the biomass, it seemed to be the same, but the biodiversity was the opposite.
Yeah. So biodiversity, it was like, with every disturbance, you have a chance to bring in some individuals of a new species into the system, but just some. So you have more individuals, more species, but also always just a few individuals, leading to lower biomass, but more diversity. Whereas if you have always strong output of the system, you kill more than you bring into the system.
Okay, sure. So fewer colonization.
Yep.
Right, cool, great. All right, our last speaker for this session is Marjolaine Stek, who's going to talk to us about nothing will stay except for change. how to manage for resilient ecosystems.
Welcome everybody. First I want to say that I finished my thesis 1.5 years ago, and there are some books at the front by the door. So there is more in depth some of the results that I have not time to talk about now. But it has the same subject, how to manage for resilient ecosystems. And I think the previous talks were a very nice introduction of this presentation, and I think there is one important thing you should notice, that if you talk about managing for resilient ecosystems, you're talking about nature managers, and they think quite different than we do, and they have other targets. So we had a good discussion about functionalities. Well, many of those functionalities are not important for managers. They think in a different way. And till recently, they had a command and a control management. So they wanted to keep what they had. Why do I say till recently? Because things are changing. And the concept of resilience is very important in this change, because it is a bridging concept. It helps to talk between different disciplines about where the world is going to, and that we are changing, how to cope with that. Well, here you see some pictures that tell something about this command and control, because it says, well, this is a nature area, but you are not allowed to go in there, because those managers are afraid that we would disturb maybe, or this nature, and then they lose their target, their goals. So they are not in control anymore. And that is also what policy is asking them, because they are paid actually for the targets they have in their area, the number of species or the species that are belonging to these ecosystems. But unfortunately, our environment is unpredictable. And on this sheet, I only show disturbances that belong to those ecosystems, but even them are not welcome. We don't want our areas to burn, because we have to start over again. And flooding is also one of these disturbances that actually belong to certain ecosystems that are not always welcome. Well, and Petersons have a very nice picture how our ecosystems can cope with those disturbances. There is, on the y-axis, there is time, and on the x-axis, space. And he shows that there are small disturbances, like, for instance, in a forest, a mouse that eats an ahorn every night. So it's a small disturbances, but it happens often. But there are also disturbances like a fire in the middle. It's quite big. It is a heavy disturbance with a heavy impact, but it does not happen that often. And we have to cope with these kinds of systems. So we have to keep in mind that this happens. So it is unpredictable. And resilience, the response of our ecosystems, can help to find a management that can cope with unpredictability. And I use resilience, the capacity of an ecosystem to absorb disturbances, organize, and maintain its adaptive capacity. For me, the function of the ecosystem is maintain its adaptive capacity. How do ecosystems react to changing conditions? For that, I used a functional approach, because ecosystems are very complex, and only the species don't tell what is happening in that ecosystems, but those traits can. And actually, I did almost exactly the same as the presentation about the dry grasslands, but I used another ecosystem, and I used other traits. And what I did, I focused on two properties of resilience, species resistance to disturbance and the species recovery, and I translated those in the traits. For instance, on the left-hand side, resistance traits. If you are a big tree, you can cope better. You are more resistant to grazing than when you are a small tree. And an example of recovery traits are many of those seed traits, because if you are eaten, you have to recover again. So you can do that by seeds, by spreading them. You can spread a big seed. It will not come that far, mostly, or it can be spread by birds. But you can also produce many seeds that go by the wind, and you are a long-distance dispersal. So there are many strategies to cope with disturbances in an ecosystem. So species know how to do it. Well, what I did in my research is one of the examples is a floodplain, and the river can flood sometimes the floodplain, and when you are close to the river, you will be flooded more often and more heavily than when you are more far away from this river. So I had information of databases about the species in the floodplain, and also about those disturbances that are part of the system. So the frequency of flooding, the strength of the flooding, those kind of things. And I had also from the databases like LEDA information about the traits that belong to the species, and then I used an RLQ analysis, and then you are going to combine 3 tables with information to get your results. And the results tell you something about the relationship between your trait and your environment. And then you can find out if in your ecosystem species develops strategies. And in a floodplain, species developed very strong strategies, like they are an invader or an avoider or an endurer. So they can withstand the disturbance, but if they get disturbed, if they are dead, they have very much difficulties to recover. And other plant species need the disturbance because, for instance, the water spread their seeds. Yep. So if you have the information about these mechanisms, you can go to the manager, and this is an example of a landscape. And you can see a lot of butterflies. So you can tell something about how you can manage this landscape in a resilient way for all those different butterflies, because they have all different wishes about what they eat, where they put their eggs, those kind of things. So you can think from small to big on a temporal scale. But yeah, I talk now about disturbances that belong to the ecosystem, but there are many more stressors in the world that we can't do anything about. For instance, climate change. So at this moment, I'm going to work on we call that the big waters in the Netherlands. So that is the Waddenzee, the North Sea, the big rivers, and how they fit in the safe operating space, and how we can use this concept of resilience to tell more of how all the users in this system, that's bigger than the ecosystem, how they can use it on a sustainable way. And we hope that resilience can help in the way of thinking, but also in managing the world actually. So this was my talk. Time for questions.
Thank you. Thanks very much. Any questions? I've got one. So, have you worked with managers and sort of local community organisation groups with some of this work?
Well, not local communities groups, but well, by accident, I work also for an NGO in nature development. So I know both parts of the story. Yeah.
And how well accepted or embraced are these sorts of ideas?
Well, we do see, at least in the Netherlands, but I think everywhere, that resilience is everywhere now. Everybody uses, but they are struggling with how can we use it, because there are so many ideas about what it should be. And I find out that it is still difficult for managers to accept that their ecosystems are dynamic and that disturbances should happen, because we actually know that they are good for the ecosystem, except for these for those stressors that play a role as well. But that you will lose some species, and we are not used to that. The policy is not used to that you can lose species. So we have to change the whole way of thinking and managing and financing this sector. Yeah, but we should also think broader or than only our own area, because when you have a big fire in your area, it is possible that species from other areas can come to you, but we often see that those areas are from other nature organizations, and they don't talk with each other, though we are talking about a network in a highly populated area, so there is it's not well connected, but we could help each other and think about, well, you have this, I have that, and sometimes those can help. Yeah.
Thanks. We've got another question at the back.
Slightly linked to your very last point, but site managers are often driven by relatively short-term conservation objectives for their site, or performance indicators, or indicators of success for their site. Does your work help to tell us what sorts of indicators ought to be chosen to describe resilience of a site that gets you away from specific habitats and species?
Yeah, by knowing how your ecosystem actually developed, so what kind of strategies you have in your ecosystem, you can also find out what you need to support those strategies. And those strategies are actually species. So if you think about, well, a butterfly is actually a good example. There are butterflies that can fly long distances. So you only need, in your area, you need to make sure that there is a suitable habitat. But there are also butterflies that can't fly that far. So how can you help them after a disturbance to recover? And those questions can help in the long term to make management decisions that can cope with a changing future. Yeah.
Any further questions? All right. Well, we might leave it there, and I have to say thank you for the speakers. That was an awesome 2 hours, and 2 hours is hard on the last day of the conference, but I was thoroughly engaged, so really great work. And thanks also for keeping time.